So the question is, are label-free going to be the future of image analysis?
You want to take that question, Paula?
Let's see. So my personal opinion is that so I think it could provide a easier and cheaper alternative. For example, for when you need applications like live cell imaging, or for example, in beating cardiomyocytes or so on. But for profiling, I hope, or I don't think it will offer the sensitivity that fluorescent microscopy is going to use. So for example, if something changes the mitochondrial morphology and that's not reflected in the bright field image feature space, like it's not we won't capture it. So yeah. Yes.
Anybody else want to comment on this?
And David, I think it depends on the resolution for one, and probably the image detection. But yeah, I think the previous speaker is right. Some of the features like beating cardiomyocytes, and I think there are probably more things that we can also grasp from bright field images. And there might be some that are not easy to detect. So I think this will shift over time as these systems get better and better. Higher resolution megapixel cameras, better lenses, and so on. So, yeah.
Okay, thank you. So a question from Barak Gilboa to Nishant. So did you use MIPs or just single planes for the conditions? Also, I guess different perturbations can have very different extent of effect on the cell population. How can you account for the extent of the effect in your analysis?
Right. Regarding the first question about planes, yes, we used a single plane for the fluorescent images. For the bright field, we used 3 planes. And regarding the next question about the effect of perturbations, well, within a single perturbation, the effect that you see, that's like for the different chemical compounds or for ORFs and CRISPRs, that's a good thing because that's how we quantify what effects a single perturbation has. But across perturbations, it's still a very good question because when you're comparing compounds and CRISPRs, they seem to have completely different effect on the cell. The magnitude of the effect is different. So it's still an open problem whether this is, can we compare across the perturbations in a very simple way. So one of the analysis that I'd shown where I calculate correlations, assume that the effect is similar, but that might not be the case. So we have to come up with better metrics and better analysis methods. The consortium is currently working on coming up with those things. So that's a good question.
Thank you. Now a question from Philip to Shui, I suspect. So how do you deal with the astrocytes that are required in the NGN2 protocol? This would also stain in cell painting.
So the NGN2 protocol produces a very pure population of cells. So ultimately, currently we don't have the issue with a contaminating second cell type. And actually we do quite a lot to avoid it. In the future, indeed, that's where we need to head, build these more complex model systems with all the representative cell types in a well. There, I think, indeed, you have to be able to identify all these different cell types.
Thank you. Loan Vuyare for Paula. Sorry, this is now skip. Yeah. So if you tried already, how well do your models perform when transferred to represent morphology of other data sets, CRISPR instead of drug screens or different cell lines?
Yes, that question still needs to be answered. So these were, we still didn't test that, but I now saw fresh results from Niranj, who showed now the benchmark the deep profiler in terms of the percent reproducibility scores, and it seemed that the deep profiler were performing worse in that sense. I don't know if it was the trained network or not.
Yeah, it was using a pre-trained network. There are lots of things I guess we could still optimize. It was just a quick result that we generated, so it's possible that the results could be improved much more. Yeah.
All right, and one of the differences is that we are training. So we are training on a data set that it comes from a different cell line, because the consortium data is not available yet to train these large deep learning models. So yeah, that's also another thing to consider. I don't know how generalizable the models will be between cell lines, but yeah, we will test all of these during this pilot time.
And the question for Niranj again, regarding effects, how do you account for off-target effects? Are those curated in the MOA?
Yes, so in many cases, the off-target effects are like each compound has multiple targets. So that has to be accounted for while doing the analysis. The results that I showed was a simple analysis where the off-target effects weren't accounted for, but a more comprehensive analysis should include the off-target effects as well.
A question for Kaisa from Roman Lane. For the iterative partially supervised training you described, how do you prevent the training session from diverging due to the new prediction being incorporated in the training dataset?
Yeah, so we use 50-50 of those in domain adaptation. We use the manually annotated PC3 training data, so the source data, to keep the precision high. And also we adjust learning rate, reduce it during the iterations.
Thank you. So the questions are coming through both in the Q&A and in the chat. So I'm trying to give justice, but I can see the speakers have already answered some of them. We may have another one. Actually, there was also a raised hand if Anna is able to ask her question.
That question has been answered already.
Okay, cool. So one actually point that is really close to my heart on the sort of JUMP consortium is the fact that I think there's not just technical and analytical problems in sort of setting up the approach of repeating the same experiment in different places, but there is a big cultural thing about this. I think if you think about academic funding, etc., the possibility of setting up grants with the aim of doing the very same experiments in different places is not as easy as it should be. And so I just wanted to ask some comments, whether in order to engage in these activities, it is essential to bridge from academia to biotech and pharma, or whether there is a possibility to sort of do things in just one or the other. Can you please comment? And also the other speakers not from the JUMP consortium are welcome to share their thoughts.
Yeah, I can share my perspective from the academic side. For us, I believe that generating such a large data set and such a diverse data set was only possible because of the industry collaboration. A single lab performing such an experiment would take very long, and the amount of effort that will be required will possibly not be possible for a single research lab to put into it. So yeah, from our perspective, having the industry collaboration, bringing their expertise into all this really helped. And also the goal of this project being generating this diverse data set from different sites and all that was only possible because of so many partners joining the consortium. So for us, yeah, we believe it wouldn't have been possible without the industry collaboration.
And also from the industry point of view, I would say that for most partners, no one would have been able to generate such a data set at this scale by themselves, or it may be not only because they didn't have the knowledge, but also the manpower and also all the knowledge exchange that has been going through all the meetings. We have like all the troubleshooting with someone has a problem, then the other partners said, okay, we had the same problem with this microscope, try this and that. So I think it's, besides just the data set, having this collaboration, this pre-competitive collaboration between pharma and academia is really amazing, and it just helps everybody, and it's much more productive than if you one partner tries to do it alone.
So this is actually very close to BitBio's values, and actually we really appreciate the opportunity from Charles River to sort of present ourselves just with a view of sharing our data and sort of engaging into the community. I think culturally there's still a lot to do. So actually, I don't know if there's a place for CRO in this. What do you think, Shui?
So, here being what Paula was saying about the ability to standardize both the data collection and the sharing the data amongst all different parties is definitely a powerful route forward to get good interpretability of the data. And I think the other point was that we can't do this alone. We need that synergy between academics, with the innovation. We need the industry standards to bring things up, to the robustness and reproducibility. And then we need all the tools to be able to execute this in a timely manner.
And I think also the among the tools, the work from the broad, both in terms of a cell painting and also providing Cell Profiler, which is kind of an open source software that really challenges the previous model, have been extremely important. I wondered if Lassi Online wants to say something about the cytodata community as a whole, if he's present, and how this group of people can contribute to these standards being formed.
Oh, thanks, Davide. So yes, there's been a very interesting presentations regarding the Cytodata Society and our aims and goals. So all these works presented today and also yesterday, but then are very interesting for our community, and well, many of these presenters are also part of the community, which we try to do as well. So like a sharing ideas, sharing new methods, sharing data sets, something that the whole community can easily use, and that's a very important aspect. And I'm very happy to also see on these presentations like the JumpCP and Kaisa's presentation that the data sets that they are using to train the models are available for the whole community. So that's really important for the coming researchers in the field.
So for me, I'd like to really thank all the speakers on the panel and Mark for chairing. We will break